My name is Ross Macklin, and this is my colleague Bill Brazier. We're the directors of Triturus Environmental, and we're a specialist aquatic and fisheries consultancy. So we spend our lives studying the fresh waters of Ireland, and we know a little bit about crayfish.
Yeah, we try our best anyway.
So today we're doing a talk on crayfish plague and aquatic biosecurity in light of the threat of crayfish plague nationally, which has spread extensively over the last 10 years and continues to spread. And we're going to discuss many of the mechanisms.
We will. First, we'll give you because we're conscious that not everyone knows much about white-clawed crayfish or native crayfish because they're fairly cryptic species. Most people have never even seen one. So we'll introduce crayfish, and then we'll kind of highlight maybe why they're so important in terms of ecology in Irish waters, including the lake here, of course. And then we'll go on to plague, and we'll explain what crayfish plague is, why it's a problem, where it's come from, and ultimately what can we do as users of waterways, what can we do to help prevent the spread, and general biosecurity measures, not just for crayfish plague, but for other nasties as well, invasive species in general. So yeah, as I said, white-clawed crayfish, they're our only native crayfish, and Ireland actually has, without doubt, Ireland has the best populations of crayfish remaining in Europe now, and that's because of plague, because plague has wiped them out pretty much everywhere else in all the other countries. Crayfish are very much found in waters that are rich in calcium because they need it for their shells, for their cuticles. And they live in a wide variety of habitats, really, from little tiny streams that you jump across, you wouldn't think there'd be anything in it, to great big lakes like Lough Oul and the likes of Derravara and places like that, mostly in the Irish Midlands. So for those of you that haven't seen them, little crayfish are probably about this size on average. They're quite small, they're not particularly big. And they're, as I said, they're cryptic. Most people haven't seen them because they're mostly nocturnal. You will see them easily at night with a torch in the margins. But you very, very rarely see them during the day. Depending on the age and the sex, mature males molt twice a year, and females molt only once in late summer. Juveniles may molt several times, so they're crustaceans. So they like a crab on the shoreline in the sea. They grow by popping off their old shell and growing a new one. So they don't just get larger, they have to physically pop their shell off. So they molt, and that's of course when they're highly susceptible to getting eaten by birds and otters and fish as well.
So basically, the NBDC has maps of crayfish distribution in Ireland, which is the National Biodiversity Data Centre. So this shows, and you can see that there's a concentration of crayfish in the limestone areas. You don't get as much when you go into the southwest, far west, northwest. Obviously, when you go into silicious catchments, so there's a strong association between alkaline limestone areas because they need obviously that for the precipitation of their shell. So when calcium carbonate goes up, as you know, Lough Oul is a rich mesotrophic limestone lake. They have plenty of calcium to grow, which is a really important component. But in terms of the long-term and recent declines, this has resulted in the crayfish being an Annex 2 species under the EU Habitats Directive and globally red listed, and it's also protected under the provisions of the Irish Wildlife Act. So Ireland is considered a stronghold in Western Europe. As you can see, this was, comprises of recent and historical distributions, and the range construction, as you'll see later, is very significant. So you can just get an idea of the spread now, but we'll show you like the prevalence of plague in Ireland since its official recognition in 2015, albeit there were earlier outbreaks in the Boyne and Whitelock, which were the initial detections of plague in Ireland. But since the advent of molecular techniques and the Fish Health Screening Unit, in the MI, they were able to, develop a surveillance program, which we'll be talking about later. So Loch Owl probably has the largest remaining lake population. So
without doubt,
yeah, without doubt, because the majority of the Irish populations would be in rivers and streams. But where you get really strong alkaline lakes, which characteristically have this type of vegetation called stonewort, chara. So you see that like what people would call that green, a structured looking weed that grows all along the bed of Loch Owl. That's a real sign of alkaline lakes, and it's actually an Annex 1 habitat in itself. We have chara communities. So like Loch Owl, for example, would be, an Annex 1 lake for a mesotrophic lake of hard waters with chara vegetation, which is an Annex 1 habitat. So the fact that Loch Owl is actually designated an SAC as well for both crayfish and as a hard water chara lake, there is a linkage between the two species. So when you actually lose a species as well, like crayfish, if you were to lose crayfish, there would be repercussions for the wider ecology, which we're going to talk about later.
You might look at this distribution map and you might think, oh sure, there's crayfish everywhere. In Ireland, an awful lot of these are historical records.
Yeah.
So like in the 80s, early 90s, pre-plague, basically pre-crayfish plague. So an awful lot of these are 10k grid squares, 10 kilometre grid squares. A lot of those wouldn't have any crayfish populations left anymore, unfortunately. So, but we'll get onto that in a bit. So yeah, just the background on why are crayfish important? Why should we care that their populations are collapsing all over the country? They're very important for the recycling of nutrients. So crayfish they're omnivorous. They kind of eat pretty much everything, but they're mostly detritivores. So they break down organic matter, mostly plant matter. So obviously that's a basic fundamental thing in freshwater lakes and rivers is you need something to break down all the leaf litter and decaying plant material. And they're also a very valuable food source for many fish species, including pike, large trout, such as especially in rivers like rich alkaline rivers like the River Shore, for example, they'd be highly dependent on large crayfish.
Yeah, and actually as well, just on that note, a lot of anglers on the shore would report when they've caught big trout, like in the Nire and other rivers like that, they were full of crayfish, or they were coughing up crayfish. Yeah. So, the implications of loss would be wide ranging, but they're a very important diet of very large trout.
Yeah, and we do a lot of work on otters as well, actually, especially in the last few years. And otters there's very much a strong relationship, a very strong correlation between healthy otter populations and healthy crayfish populations. So if you have lots of crayfish, they're very easy for an otter to catch. Obviously, crayfish are hiding under rocks most of the time, so an otter, it's candy from a baby kind of thing. Yeah,
and you can see here, this actually plate here is actually where the otters have predated on the carapaces and the heads of crayfish. So you can see that they kind of take a bite out and they leave the rest of the parts. And then also their spraying sites, which we would call piles of otter spraying, we call them like midden sites, because they're actually full of shell remains. And like if you go on to important crayfish rivers like the Liffey, for example, you'll see underneath bridges where there's actually piles of otter sprained with just the remains of crayfish left behind. So you can actually tell as well, whether there's crayfish in a river site by looking at the presence of crayfish in otter remains. So sometimes you mightn't find crayfish, but you look under a bridge and you see the crayfish remains in the otter spraint, and there's crayfish somewhere in the catchment. And if you look at the range of otter, for example, if a female otter would have a tighter territory than a male, say circa 5 kilometres, and a male can be 20 kilometres and above. So you could then draw a radius and you could say, well, it's likely to be within this kind of range of that area, and that can actually help you if you're trying to figure out where crayfish populations are.
Yeah, so the threats to crayfish, obviously they were threatened before crayfish plague came in less than 10 years ago when it got really bad. So as you could substitute crayfish, you could use almost any species, unfortunately, native species, and say much the same thing as habitat loss is a huge problem. Pollution from stormwater and wastewater treatment plants not efficiently working, things like that. Crayfish, because they're quite easy to catch, in traps, very easy to catch a baited trap. There's been a lot of illegal fishing, poaching and stuff like that, especially on the canals actually. It's been quite a major problem. The kind of the big threat, which is quite peculiar, and we'll talk about it later, is as we said, white-clawed crayfish are our only native species, but I was going to say we don't have any non-native crayfish species. We do have one now in one location, but it's quite unusual in a European context. In fact, every European country has multiple non-native crayfish species as well as a native species. Ireland is quite unique in that respect.
Yeah, we'll be talking about it in a minute. So like in terms of like, there's some of any nicks in the room though?
Nicks.
There's no nicks.
No nicks.
Well, this is the bad science joke. Non-indigenous crayfish species are known as nicks. So basically some of the, there's a lot of potential, I suppose, invaders. Like we've always talked about the signal crayfish, which is this gentleman here with the large red claws, which a lot of people have purported in Ireland at times because they see sometimes the claws of white-clawed crayfish can look orange, reddish, at certain times of the year, especially when they're under stress. And then someone has reported, because when you think of white-clawed crayfish, they think, but their claws aren't white. It's actually white patches underneath the claws, pale patches, not even white patches. So it's been a bit of a confusion with regards to the common name. So you also have like the marbled crayfish here, and that actually can, under certain conditions, reproduce itself. It can fertilize its own eggs, which is really interesting. So even if there's not just males or females, it can reproduce. So there's also the red swamp crayfish and the spiny cheek crayfish, and obviously the red swamp crayfish is bright red, and the spiny crayfish has these iron tips, bands, and spiny cheeks. So but we haven't detected those particular species in Ireland as yet.
No.
And of course, the main threat is the crux of our talk here, the crayfish plague, which we'll be talking about in more detail. And actually one non-indigenous crayfish species, which we haven't mentioned here, which is actually present in Ireland now.
Yes.
But we'll talk about that in a minute.
Yeah, we'll go on to it in the next slide anyway. But most nicks for actually carry crayfish plague. They might not be necessarily impacted by plague themselves, but they carry it. So that's why it's such a concern to try and prevent them coming into Ireland. But now we have crayfish plague anyway. So crayfish plague, what is crayfish plague? It's a lot of people call it a fungus, but it's not. It's technically a water mold that produces spores, and it's just very, very deadly to crayfish. They just can't deal with the infection. It can cause a lot of people say it causes 100% mortality in crayfish. It can cause 100% mortality in the population, but not always. So it's not a case of, well, you have crayfish plague, you have absolutely no crayfish left. You can have some crayfish remaining after the infection, which we'll get on to. Crayfish plague originates in North American species, which were originally brought to Europe for food production in the 1800s and early 1900s. And that's where pretty much all the non-native crayfish have come from. By the way, in Europe, I think there's like 20-something different species of non-native crayfish species in Europe now. They all came in for food production, and they all escaped from cages and farms. And nearly all of them brought plague with them. So white-clawed crayfish used to be across much of Europe, and now they're not because of crayfish plague.
It spreads, so it produces these mycelium and hyphae masses, and you have a spore which comes out through the cuticle, and it bursts and spreads.
Cuticle is the shell.
So the cuticle is the shell. And so first of all, the mechanism of how it kills, because I think it's important, people often ask that. So basically when it attaches as a cyst, it detects a suitable surface, and then the hyphae grow in through the cuticle and into the body, into the tissue of the crayfish, and that just kills. So it basically invades all of the organs, and eventually the crayfish just dies. So some crayfish species, like the North American crayfish, signal crayfish, are resistant, and they don't die from the plague. So they can be a carrier and survive, while other crayfish species like our native crayfish, Ostropod mobius pallips, the white-clawed crayfish, suffers a different fate because the mortality is generally very high, and there's multiple strains as well of plague, which we'll talk about a bit in a minute. We won't go into that. But so the hyphae grow, and then the hyphal mass bursts, and the zoospores, which are these dinoflagellates, swim and they find an appropriate surface. If they have a surface which isn't recognised as say a crayfish or something, they can invade, or even other crustaceans, because there is other, not just crayfish are susceptible to actual carrying plague. It will burst again and swim again. It can do actually up to 3 cycles. So that shows that it can have an indefinite persistence, you know, for long periods of time without a host. So it could attach to a surface, insist, and then burst again, swim till it looks for something, then cyst again, and so forth, up to 3 or 4 times. Now there's more research needed on this, because you can imagine where it's been studied inside in laboratories tells you so much, but in the wild it could behave completely differently. So this tells you that it's very, very capable of transportation, you know, through water bodies and actually attached to substrate. So for you know, anglers with waders, kayakers, you know, any water, wetsuits, dry suits, you know, it could potentially attach, or else be in for short periods of time, or particularly if you have any water, if you're carrying any water in the bottom of a boat or an impeller. You know, an impeller in a water-cooled engine is going to suck the water in, and it could stay in that. And then you move that into a new lake, you start the engine, and the impeller will flush out the water from the other water body.
Yeah, so it spreads by spores, and you think in evolutionary terms, you know, nature evolved, or things evolved spores are designed to be very resistant to adverse conditions, so they're designed to be dormant for long periods of time. I think the research says that crayfish plague spores are viable for 2 or 3 weeks in water, but there hasn't been a whole lot of research into the dormancy period of the spores, which can probably be much, much longer. So it's another reason why crayfish plague is has been so prevalent across Europe, is because you know, the spores can settle in the sediment at the bottom of a river or lake, for example, and possibly you know, reactivate at a later date, as spores do, right? And
we don't know, they could potentially attach to other, like mollusk species, you know, or even crayfish species, to shells. I mean,
yes,
there's it's difficult because you have to really figure out what it's doing in the natural environment, not just in laboratories, to see exactly what's going on.
So contaminated water, whether that's on a boot or in a boat or in a bucket or on a net or a bird's feathers or an otter's fur, it can be spread a multitude of ways, and that's one of the main problems with it. Other crustaceans, as we touched on a while ago, other crustaceans, there's a definite lack of research on this, but we do know there's a certain couple of species that absolutely are carriers for crayfish plague. There's one, the main one being Chinese mitten crab, which we do actually have in Ireland. This is a picture we got from a survey we did in the Waterford Estuary last year. So we have Chinese mitten crab in Ireland that came in, 15 years ago or something. The first one was recorded. They haven't really taken off, which is a bit strange. But if crayfish plague can infect Chinese mitten crabs, then it stands to reason they can probably infect other species in Ireland, other crustacean species, for example.
And the other thing about these guys is they actually move from estuaries in towards freshwater. So they actually migrate from the transitional parts of river systems, like and they move up into the freshwater reaches. So if you think if it was a carrier, it could potentially take it from an estuarine environment into a freshwater environment and then moved further on. So, you know, when you think about...
And they also walk across land between rivers too, so they can go from one catchment across to the other. It's crazy. Chinese mitten crabs are a huge problem in England, southwest England, or
Holland as well.
Southeast England, but not here. But despite what I said about, you know, any kind of water can spread at birds or otters, there's even been some research looking at whether crayfish plague spores can travel through the guts of fish and mammals, come out the other end and still be viable. The jury's out whether or not they're still viable. But irrespective of that, humans are the #1 vector for most invasive species, and this is no exception. We said earlier the non-native. We do have one non-native species of crayfish now, but it's confined to one lake. It's an Australian species, so it's called the yabby, Chirax destructor.
And you can tell by obviously the species name, the destructor part, that it's probably not great for the environment.
No.
But this was Pascal Sweeney, discovered this population in Cork, and you can just see the very strong, robust claws, more like a lobster claws than a
They're
than
more
a
like a
crayfish
freshwater
claws.
lobster, really.
Yeah.
Aren't they? They're huge. I mean, compared to our crayfish, they're massive.
And interesting as well about this Chirax destructor, like from what I know currently, I don't believe that there's a breeding population in the UK.
No.
But we have it here, and they have all the species that we don't have. It's kind of interesting that this one got in here. So this was likely an aquarium trade escapee, and it's in a quarry lake, so it's self-contained. And these are known to walk across land as well. But the population is breeding in the Irish temperate climate. A lot of people talk about temperature restrictions for breeding of invasive species in Ireland, but you can get adaptation from ornamental species, and they can they can do things that they're not supposed to do. Let's say.
Yes. The history. So that's the theory behind it. The history of plague in Ireland is very, very recent. It was considered largely plague-free until 2017, although there was a very kind of obvious mass die-off of crayfish in the Boyne catchment in the 80s. Mid-80s. The genetic techniques were in their infancy then, so it wasn't really detected. Everyone kind of knew it was probably crayfish plague, but we didn't actually prove it was crayfish plague. And then again, there was an isolated incident in a small river system in Cavan in 2015 where there was plague outbreak, which was tested and confirmed as plague. But things didn't really start to go wrong until 2017, and then suddenly it popped up in the Shore, and then the Barrow, and then the Deal in Limerick, the Lorha at the top of Lough Derg, and after that, pretty much, as we'll see in a minute, pretty much every major catchment in Ireland, it spread rapidly. So in response to this, the MI, the Marine Institute and the National Parks set up a collaboration and set up the National Crayfish Plague Surveillance Programme, which has since then done nationwide testing using genetic techniques to try and figure out, well, where is the plague and where are the remaining crayfish populations. You asked earlier on, or is there any way of detecting the spores of crayfish, and you see them and you can't, they're microscopic, and they're very, very small. So that's where genetic techniques came in. The question was asked, well, how are we going to know where this stuff is? And that's where something called environmental DNA or eDNA came in, which is basically every living organism, plant or animal, releases genetic material, DNA, into the water, into the air also as well, and the soil, but in this case the water. And we can now detect that, which is just amazing, really. We do it all the time now for work routinely. In pretty much every project we do, we use eDNA for various species, crayfish included. But so the MI and the MPWS did, have since 2018 have been doing this nationwide testing, and very quickly they realised that it was in far more places than we thought. So obviously we had the original outbreaks on, the Barrow, for example. I think there was like 400,000 dead crayfish, something like that. It was like just pretty much every single crayfish, pretty much all the way down the River Barrow in a matter of months, just dead, gone.
And the same in the Shore. And
the same in the Shore, and pretty much everywhere else as well. It starts 2015, 2016 with the Brewski River. These are river catchments, by the way. They're not individual sites because there'd be too many points on the map to show. But you can see the spread of plague in such a short period of time. This is all with eDNA detection, basically. So eDNA detection of plague. The red is the Marine Institute and MPWS collaboration. You can see the spread has been so quickly, but notice the yellow at the end. The yellow is our data, which we've got in our various tests. These are the yellow is catchments where we've detected plague that the MI haven't sampled. Now, the MI haven't sampled those primarily because there's no crayfish populations there. Yeah. So
but we have plague.
So, for example, the Mal Bay catchment here in West Clare, number 28, or the Lee in Cork, where we're from, have never had white-clawed crayfish ever.
And yet we have plague.
And
so what's going on
there's
there?
plague all over the catchment, which links back to, obviously there's many things that can spread plague, but also to us at least, it kind of implies that plague is able to survive on something else other than crayfish. Yes, it makes logical sense. And that's going to have to be tested.
And if it's that widespread, you know, how much plague is there really out there? Is it every catchment in Ireland? You know, because we're talking, you know, you're seeing here a very radical, expansion, and you know, a lot of these records may even been here before we started the testing program was even started.
Yes, quite possibly.
You know, there was speculation. I know I often talk to Pascal Sweeney, and he speculated that you know, in the early days, I think the early 2000s, that there was plague already in the north. Yes. So like, you know, but to validate these things, you have to do it, you know, the proper way through proper health screening. Yeah. And you can see most recently here 18. That was the Blackwater outbreak this year. Again, I think Pascal was the first person to spot that. And when we checked some of the areas that had crayfish, for example in Fermoy, you know, in the centre of the town, there's no longer crayfish present. And also, like, you know, when we went back to 2020, we were finding crayfish, you know, under all the stones underneath the bridge and near the weir. And the crayfish was in all of the otter spraints. And when we went back this year, the crayfish were gone, and there was no crayfish remains in any of the otter spraints. Yeah. And interestingly, we got a great video of an otter and the way he was foraging, and he was on the bottom of the gravel island downstream of Fermoy, and he was turning over all the stones and digging his head into the stones, and with his back legs he was kicking up all the rocks and stones and actively searching, but he was actually catching fish. So we were thinking that it was a behavioural technique he'd adapted for crayfish that he was now applying to catch all, and he was doing quite well, because every couple of minutes he was bringing up a fish. But it just shows that crayfish are really important, but some species that are very recalcitrant, like otter, because otter populations have been expanding in Ireland, may still survive, but it may have local population changes may happen, and other species may be more greatly affected. But again, we don't have enough information at this stage to say what are the implications of a nation-wide spread of plague on really important species for biodiversity like otter, you know, which are also incidentally an annexed species, a qualifying interest of many of the SACs in Ireland. So we lose crayfish, which is a qualifying interest species, and we may impact otter, which is also a qualifying interest species. So you can see that the implications of a loss of a keystone species like crayfish may have huge implications for other species as well.
Absolutely, yeah. Just before we get off the map, you'll notice that this is where we are. We're up here somewhere. There hasn't been plague detected around this area yet. Yes, so other than detecting it by eDNA, as Ross was saying, it's a great way of getting a heads up on whether there's plague in a catchment or in a river or in a lake. But on the ground, because not everyone has eDNA kits, of course, on the ground, how would you recognize a crayfish plague outbreak? Well, it's fairly obvious, you know, without sounding derogatory. You will see mass mortalities of crayfish. You will likely, because it acts so quickly in a matter of days, certainly within a couple of weeks of infection, you will have an awful lot of dead crayfish. The infection, as we were describing earlier, because the hyphae, which a fancy word for root system essentially, because it penetrates into the internals of the crayfish, it impacts their organs and gives them a pretty horrible death, I would imagine. So one of the main impacts it has is it impairs their vision. So crayfish are, as I said, they're not largely nocturnal. But when they can't see, they literally can't see if it's day or night. So if you see crayfish crawling around during the daytime in really, really shallow water, they're doing that because they can't see. Obviously, a crayfish would not naturally do that because it's going to get eaten by something, right, like a heron or something. And they're generally they just have this malaise about them. They're slow and they're lethargic, and you can tell they're sick. So it's fairly obvious when a plague outbreak happens. The hyphae, the fungal-like mass on the outside of the crayfish, you'll see they have this like fuzzy appearance, this white fungus essentially on the outside of them. That's a classic sign. And another classic sign is melanization, which is basically dark spots. I know you probably can't see it very well there, but you get dark, obvious, conspicuous spots on the crayfish, on the outside of the crayfish. It's another classic sign of crayfish plague. They do lose limbs and stuff like that because it's impacting all their cells and their whole internal structure. But crayfish lose limbs all the time, by the way. They fight with each other for territory and stuff. So you'll almost always find lots of crayfish with only one claw. So that's not a good diagnostic characteristic. So it comes back to basically genetic analysis is needed to be absent. And earlier as well, so a latent infection, latency is just a delayed infection basically. We'll get into it in a second. There's various, like any virus or infection, there's various strains. Some are stronger than others, some are more persistent than others. So if you have a particular strain of crayfish plague that isn't very virulent, you could have a latent infection. So it might not immediately kill all the crayfish. It could take a few months to weaken their immune system and then cause a kill. Or conversely, it could be literally within 3 days if it's one of the really strong strains of plague. What they're finding in a few European countries, so Galicia, so Spain and a bit of Portugal, and also in Finland,
France,
I think.
France as well.
And possibly France. They are definitely picking up signs of natural resistance to plague. So there's been plague outbreaks for 20, 30, 40, 50 years, and now they're starting to see that some crayfish populations are actually recovering, white-clawed crayfish populations.
And the MI have found the same in Ireland, like, preliminary research, they found some evidence of some resistance developing.
Yeah, well, we find it quite often, don't we? Where we test, so we routinely test, if we're testing for white-clawed crayfish, we also just test for plague at the same time. And we're finding more and more that there are now several catchments where you do have crayfish and you do have plague in the same place. So that would also imply, it's not definitive, of course, but it would all imply that there's some level of natural resistance.
And we're also seeing some juvenile crayfish coming through.
Yes. Yeah, exactly.
That we do find them in sites that have had plague outbreaks. Like we found them in the Barrow there recently in a Duishka. Yes. Like where there was crayfish plague and there was no crayfish. Yeah. And this year we found crayfish. Yes. Again, small population, but
Yeah. So as water users, whether you're an angler or a boater or a swimmer or whatever, it's aquatic invasives and aquatic pathogens are very hard to stop, very, very easy to spread and very hard to stop once they have spread. So it's just an example here of the check, clean, dry approach, which is kind of like standard best practice for biosecurity. So whether you have your boots, or I suppose it would even extend to your dog who's been in for a swim or something, you check them, make sure it's
work on that dog.
Yeah. Hose him down.
Dry him out in the sun.
Yeah. Bad example perhaps, but you get where I'm going.
But actually, it just shows you that there's a lot of risk. You know what I mean? You think things you don't even think of. Yeah. Maybe your dog jumped into Loch Owel and he runs home and he jumps into the ditch, and then that ditch is connected to a river. So like sometimes, but I think the thing is there's a lot of stuff which, is controllable and some stuff that is out of control. Yeah. And I think the one thing what you got to remember, it's trying to, it's not just preventing the spread, but it's the pace of the spread. If you suddenly say, let's just forget about everything, we let it go wherever it goes, no rules, no anything, it would be a lot worse. So and the other reason that's a problem is if you release something and spread it rapidly, the native fauna don't get enough time to acclimatize to the actual changes. Whereas sometimes if you get slower release and these invasive propagules, as we call them, the lag time between the point of invasion and the point of establish is much slower. That allows nature a lot more time to get accustomed to that change. And in many ways you can see that through crayfish resistance and through the different strains, that if you get a less virulent strain that comes in and then the crayfish actually don't all get wiped out by that strain, but they're starting to get used to it, and then another strain comes in, they're building up immunity over time. It takes a lot of time, but so if you're doing this rapid spread, it's much worse. But and also, everyone has the duty of care to actually think about, what they're doing and where they're going. And it's not just catchments which are connected to rivers, but these isolated places. I think, you know, isolated lakes that get very, very little human activity, they're particularly important, because they're going to be the places that are going to be the last areas untouched. And there is some of them in this county, you know, like that are really inaccessible lakes across tons of fields, and no one really goes into them. And I think they're the places as well that we need to be especially careful, because they might be the marker sites that have the most pristine assemblages of flora and fauna that are untouched. So I think education is really important as well, and this is why we're doing this, so people actually know mechanisms to actually minimize risk. These are some of the ways actually just to you know, to disinfect and clean your equipment. So you can see here, like, the plague could be spread by crayfish fragments excreted by fish, but unlikely birds or mammals. We spoke about this earlier, that you know, it doesn't survive well through the digestive tract. There is some evidence that maybe the cysts could stay on the skin of fish, but not for a long period of time, because they don't survive well. But that also depends on the time. So, like, for example, I'll be talking a bit about a stocking, and you've, say, crayfish plague attaches to a fish. You take the fish from another place, you put it into your lake, or even with the water, for example. But even without the water, could it survive on fish? Now, the risk is typically low, but it's just to think about that. We don't know yet. I mean, like, the evidence is suggesting that it doesn't pass through the gastrointestinal tracts of, like, say, for example, otter that might prey on a crayfish in one catchment that has plague and then spread it into another catchment. These things require more study. And in terms of spores, we talked about the viability of them as cysts for several weeks in water. That's a long time. So these spores are really tough. So you know, the check, clean, dry approach is the most commonly spoken about technique for you know, ensuring that you minimize the risk of spreading invasive pathogens or invasive species. So dip, one of the most important things is, like, you really want 15 minutes not dipping. So it's you
see this a lot,
period.
don't you? We see it all the time, don't you? In loads of different groups. They have a bucket of disinfectant, they're doing everything that they're supposed to do, and they get their contaminated material and they literally go like that. It's useless. It doesn't do anything. It's not long enough. It has to soak for at least 15 minutes.
And just one thing I wanted to mention here as well is the heat, the steam washing, right? I mean, one thing about machines, right? Machines have been, you know, implicated in spread of plague, because, you know, you're digging out ditches or drains, or maybe landowners have a machine, or even doing riverine works. You should really be steam washing the machine, you know, like outside of drying. You know, like 60 degrees, you can get steam washers. You steam wash the track wheels, steam wash the bucket, anything that's had contact with water, and then you allow the machine to dry out before transferring it into a new catchment. Virkon, really, you know, you're only doing that if you're in a difficult situation and something has to move. But ideally, you never want to be moving between catchments without steam washing and drying. But the Virkon dip can be effective for people who, you know, maybe they're working and they just want to, you know, reduce risk. We'll just talk briefly about this. I think this is an important one just for stockings, you know. I understand, like Lochul gets stocked with triploid trout, doesn't it, you know, so they don't breed, obviously, with the native brown trout population. But just something, you know, obviously we're talking about the risk, but, you know, this Oidman paper, you know, specified the fish movements from a site of a potential crayfish epidemic or area of plague should be generally avoided. And they just mentioned that spores can be transported under the water, as we discussed, in the mycelium, spores of mycelium on or in the skin of fish, right? Even though survival probably isn't very long. Mycelium and spores in the gastrointestinal tract, the gut of fish, although unlikely, there's still more research required. And crayfish themselves transported accidentally with fish. So say if the Charax structure spread out and it was in another area, and, you know, a fish eats it, or maybe it was half alive, or, you know, maybe it was in the tank of water, you know, it could potentially be transported into the new water body. So, you know, we're saying, like, you know, obviously, I don't think fish are fed before transport anyway, just to clear out the gut. Disinfect the water to ensure, you know, to make sure, I mean, if you're using mains water, a lot of the time the mains water is you know, chlorinated and disinfected anyway. So if that's sitting there, it's probably the best water to transport fish in, if it's coming off the mains rather than coming up from a river or a tank. And then, you know, just be mindful of the water source area where the fish are coming from. Is there plague in that area?
So yeah, what do you do if you suspect an outbreak of crayfish plague? What can you actually do? Well, definitely report it straight away. If you even if you have a thought for a second that, oh, there's something wrong here, definitely report it. Don't just say, oh, sure, it's probably fine. So report it to the National Parks and IFI. They're the two main ones. The Marine Institute would probably be notified anyway. The Marine Institute, by the way, the reason they're involved in all this is they have what's called a fish health unit. They do all the testing for fish and shellfish diseases in the country. That's their job, so that's why they're involved, even though they're called the Marine Institute. They do an awful lot of freshwater work as well. Of course, the same with any invasive species, whether it's plague or a plant or anything, be as specific as you can with details of the location. You know, what did you see? Where exactly did you see it? Can you send a pin? Can you pin it on Google Maps, or take close-up pictures or a video? You know, which I think most people would know to do in this, you know, this day and age. Anyway, and yeah, like I suppose, like we were saying, this is where the whole idea of local trusts and local biodiversity groups are coming from. I mean, they're spreading up all over the country now because of the same issues we've kind of talked about today. It's like, oh, well, what do we do if this happens, or whose responsibility is this? I suppose, you know, raising awareness locally. You need to work together, really, the local stakeholders. I mean, I'm not trying to put the onus on anyone in particular, but that's how these things are going to be resolved, I suppose, is the people working together. The people who really care about the lake, you know, for whatever reason, they need to work together for the protection of the lake. It's all well and good to think that it will come from the top down, and sometimes it does, and when it does, it's great, but it often doesn't, as we know. It kind of has to come from the ground up, has to come from the local stakeholders.
And the other thing is, like, you know, you know, like your mandate for, say, biosecurity management, you know, citizen science is a big thing now. It's not just us nerdy scientists here, but, you know, most, you know, members of the public can actually get actively involved in science too. Yeah. So, you know, we'll be looking into, you know, in the future, you know, at the opportunity for citizen science-based eDNA as well. You know, so you might, guys, take your own sample from your local lake. You know, maybe something that we could do in conjunction with the local authorities in LAWPRO, and then, you know, you get your results, and you know your lake is free of plague. And then there might be other things, and then, of course, if your ear is to the ground, and maybe there's new novel techniques that are coming out. You know, maybe they come up with some, you know, type of vaccination type process that is newly discovered that might be able to be rolled out, or, you know, you never know with science. Science is changing so fast, so rapidly, and obviously with the AI coming in now, you know, we're not going to believe what will be possible in the future. And I think this is something to always be aware of, that, you know, there's always novel things that come out on an annual basis. So if you're aware of those things, or if you know, commission experts like ourselves to look into those things, you know, we can often come up with great ideas for, you know, improving practices and, you know, creating management plans and frameworks that can be adopted. So, yeah, thanks a million for listening.
Yeah, thank you. Thanks for listening.
Yeah.